Hi, I'm Evan, and I'll be leading you through a study guide review of the material from Unit 1 of Anatomy and Physiology 1. Here we go. We'll begin with a review of Chapter 1, an introduction to the human body. We are studying anatomy and physiology. Anatomy refers to the study of the body's structures. Gross anatomy refers to large structures like bones, muscles, and organs, while microanatomy refers to microscopic structures like cells. Physiology is the scientific study of the chemistry and physics of the body's structures, so basically function. Recall that human bodies have 6 basic levels of organization. The smallest level is the chemical level, which consists of atoms and molecules. 2 or more atoms combine to make a molecule. Molecules combine to form cells, which remember are the smallest unit of life. Tissues are formed by groups of similar cells. 2 or more tissues combine to form organs. Organs that work together comprise organ systems, and the collective organ systems make up the organism. The different organ systems in the body have different functions and roles. They work together to form functional humans with general characteristics. First, organization. The body's cells are organized in a way that creates separate compartments within the body. Cells have a selectively permeable membrane that allows them to regulate what enters and what leaves. Another example is the blood vessels that keep blood within the cardiovascular system. Metabolism refers to the continuous building up and breaking down of molecules provided by the foods and beverages we consume. To provide the body with energy and other molecules, the body needs to form its structures and carry out its functions. Responsiveness refers to the ability of an organism to adjust to changes in its internal and external environments to maintain homeostasis. Movement not only refers to body movement, but also the movement of cells and organs within the body. Development refers to the differentiation of the body's cells, which creates specialized cells from undifferentiated cells. Growth refers to an increase in the number of cells, while reproduction refers to producing offspring. Humans can only survive under certain conditions. Oxygen is a requirement for life. It's needed for many chemical reactions in the body, including the production of ATP for energy. Nutrients from water and food are also necessary to sustain life. Water is critical for metabolism, transport of cells, nutrients and gases, temperature regulation, and insulation. Foods we consume provide us with organic compounds, vitamins, and minerals that are necessary for metabolism and other body functions. The human body can only live within a narrow range of temperatures that surround the body's set point of approximately 98.6 degrees Fahrenheit. When body temperature falls outside this range, protein structures change, which prevents them from functioning properly. Finally, the body can survive within a narrow range of atmospheric pressure. This pressure keeps blood gases dissolved and allows the proper exchange of oxygen and carbon dioxide in the lungs. Homeostasis refers to maintaining a relatively constant internal environment in the body. Homeostasis is maintained by balancing ions, pH, chemical reactions, and transport of molecules between the body's internal and external environments. Physiological processes in the body operate around set points. The body only functions properly within a narrow range of conditions above or below the set point. When conditions stray too far from a set point, dysfunction up to and including death are possible. Negative feedback systems try to prevent this. One example is fever. When a sensor detects a body temperature above normal, it sends signals to the control center to initiate a response by an effector, which here would be the sweat glands of the body opening to release heat. This action is intended to return the body's temperature to the set point. Positive feedback systems push a change further in the direction of change, but only temporarily. An example of this would be childbirth. Okay, recall the anatomical position, which is the body standing upright with feet shoulder width apart and parallel with toes facing forward and upper limbs held out to the sides of the body with palms facing forward. Also recall that anatomy is described from the body's perspective, So structures on your left as you look at a body are on the body's right, and structures on your right are on the body's left. The use of standardized directional terms in anatomy allows for the common understanding of where structures are located on the body. This slide describes body directions using an image from the text. You should be able to use these terms correctly. Another way to describe positions on the body is through the use of directional planes. The sagittal plane divides the body into left and right sides. The frontal plane divides the body into anterior and posterior, and the transverse plane divides the body horizontally into upper and lower portions. One way the body maintains its internal organization is by the separation of compartments into cavities. The dorsal body cavity contains both the cranial cavity and the vertebral cavity. The ventral body cavity contains both the thoracic and the abdominal pelvic cavities. The thoracic cavity is further divided into the mediastinum, pleural, and pericardial cavities, and the abdominal pelvic cavities contain the abdominal and the pelvic cavities. The abdominal pelvic region contains many of the body's organs. It is divided into 9 regions and 4 quadrants for clear communication. The regions and quadrants noted in this image from the text should be learned. This image indicates the names of the body regions on the anterior and posterior sides of the body. Names and locations of these regions should be learned. Imaging techniques allow healthcare workers to view internal structures of the body without opening the body surgically. X-ray images are made using high-energy electromagnetic radiation with a short wavelength that can penetrate solid structures like bones. X-rays are best for visualizing bones and teeth. In CT, or computed tomography, a scanner rotates around the patient taking X-ray images that a computer then combines into a 2D view of the area that was scanned. CT is especially good for the scanning of soft tissues like those in the body cavities. It allows doctors to measure the size of structure with precision. One drawback is that it exposes the patient to a large amount of radiation, which could put them at higher risk for cancer. PET scans use radiopharmaceuticals to produce an image. Their main advantage over the other imaging techniques is that they can show physiologic activity like blood flow. They are useful for diagnosing heart disease and the spread of cancer. Ultrasound uses high frequency sound waves to produce an echo signal that a computer converts into a real-time image. It is the least invasive of all these techniques and is often used during pregnancy. It is useful for studying blood flow, heart function, and fetal growth and development. MRI, magnetic resonance imaging, uses a large magnet to create a magnetic field that bounces radio waves off of structures, creating an image. MRI is especially useful for discovering tumors. It does not expose the patient to radiation, but is expensive and can be uncomfortable for the patient due to the loud noises made by the magnet and the confined space in which the patient must remain. Chapter 2 examines the chemical level of organization of the body. Recall that matter is anything that has mass and takes up space. The atom is the smallest unit of matter. Atoms are made up of a nucleus containing positively charged protons and uncharged neutrons, surrounded by a cloud of negatively charged electrons. Atoms are most stable when they have a full outer shell of electrons, called valence electrons, and will give up, gain, or share electrons with other atoms in order to achieve this. Bonds are attractions, either weak or strong, that hold atoms together in molecules. The 3 most common bonds are listed here. Ionic bonds are close associations between ions of opposite charge. The example here shows an ionic bond between a positively charged sodium ion and a negatively charged chloride ion. Sodium has donated an electron to chlorine, making sodium positive and chlorine negatively charged. Covalent bonds occur between atoms that share electrons. Nonpolar covalent bonds have a relatively equal sharing of electrons within the molecule. Polar covalent bonds share atoms in such a way that One end of the molecule has a slight negative charge, and the other has a slight positive charge. The water molecule seen here is an example of this. Hydrogen bonds form between two molecules like water molecules, in which one of the hydrogen atoms is already bonded to another atom with slight negative charge. Metabolism can be thought of as the sum total of all chemical reactions in the body. Reactions are either catabolic or anabolic. Synthesis reactions combine reactants into products. Energy is required and is stored in the bonds. In decomposition reactions, a larger molecule is broken down into parts, causing an energy release. Enzymes are proteins that speed up chemical reactions by lowering the activation energy that is required for the reaction to proceed. Inorganic compounds are important to life. These are compounds that do not contain hydrogen and carbon. Some examples include water, which is vital to life and serves many functions in the body, as seen here, salts, which are needed for conducting nerve impulses, digestion, and bone formation, and acids and bases, which, along with buffers, serve to balance the pH of blood and body fluids. Organic compounds contain carbon and hydrogen. Four important classes are noted here. Carbohydrates are used for fuel for the body. One metabolic product of carbohydrates is glucose, which is converted into ATP, the form of energy the body can use to carry out its processes. Proteins are made up of amino acids and are used in all body tissues. Some proteins function as enzymes in chemical reactions. Lipids are the most varied group structurally. They include triglycerides, phospholipids, and steroids, and are used as body fuel in the cell membrane structure and in signaling. Nucleic acids are made of nucleotides that contain a phosphate group, a pentose sugar, and a nitrogenous base. Nucleic acids are the genetic information in the cell, DNA and RNA. That brings us to Chapter 3, the cellular level of organization. The cell membrane is the structure that surrounds the cell, separating the contents of the cell from the outside environment. The membrane is a phospholipid bilayer. Phospholipids form the structure of the membrane, with their hydrophilic heads oriented toward the intracellular fluid and the extracellular fluid, and their hydrophobic tails between the layers. Cholesterol and proteins are dispersed throughout the membrane and aid in cell recognition and the transport of substances across the membrane. The membrane is fluid, meaning that its components have movement. Cells need to be able to communicate with their environments. Recall that the cell membrane is selectively permeable, meaning that only certain things are able to cross it freely, and other things must have help. The cell uses passive and active transport mechanisms, as well as endo and exocytosis, to accomplish this. Passive transport mechanisms allow gases and small molecules to move down their concentration gradients, and therefore no input of energy is required. A selective few gases, hydrophobic molecules, and small polar molecules like water are able to diffuse through the membrane. Charged particles or large molecules like glucose have help crossing the membrane by transmembrane proteins in a process called facilitated diffusion. Osmosis refers specifically to the movement of water into or out of the cell down its concentration gradient. Alternatively, active transport requires an input of energy to move substances against their concentration gradients. Finally, endocytosis and exocytosis move substances into or out of a cell using membrane-bound vesicles. Organelles are part of the cytoplasm. They are membrane-bound and have specialized functions. The rough endoplasmic reticulum is connected to the nuclear envelope, is dotted with ribosomes, and is responsible for making proteins. Ribosomes also exist freely in the cytoplasm. Smooth ER makes lipids and steroids. The Golgi apparatus sorts, modifies, and transports proteins coming from the rough ER. Lysosomes contain enzymes that digest materials in the cell. Mitochondria are the sites of ATP synthesis. Finally, peroxisomes contain enzymes that participate in the metabolism of lipids and chemicals. The nucleus of the cell stores the genetic information. It is enclosed in a double bilayer membrane called the nuclear envelope. This membrane contains protein pores that allow for communication and transport between the nucleus and the cytoplasm. The nucleolus, shown here in dark purple, is where the RNA that is needed to create ribosomes is made. DNA in the nucleus is stored as loose chromatin. When a cell prepares to divide, it must first replicate its DNA so that each daughter cell will be nearly genetically identical. During the initiation stage, an enzyme called helicase relaxes and separates the two strands of the double helix. During the elongation stage, the enzyme DNA polymerase reads the sequence of nucleotides on the parent strand and adds the complementary nucleotide to create a complement to each parent strand. During termination, when replication is complete, the two double helices form, each being nearly identical to the original parent DNA. Protein synthesis consists of two phases, transcription and translation. During transcription, a messenger RNA complement of the gene is made. To do this, an enzyme called RNA polymerase recognizes a DNA sequence that lies ahead of the gene and binds to it. It then initiates making an mRNA complement of the gene. Recall that there are no thymine nucleotides in RNA. Uracil will take the place of thymine. Upon termination, the mRNA detaches from the DNA and the enzyme. The mRNA molecule is modified by removing segments called introns that don't contain coding sequences. The remaining exons are then spliced together, and the strand leaves the nucleus for the cytoplasm. Once the mRNA is in the cytoplasm, two subunits of a ribosome come together on the mRNA and read its sequence, 3 bases at a time, called codons. Each codon codes for a specific amino acid. Transfer RNA containing an anticodon on one end and the corresponding amino acid on the other recognizes the codon and adds the amino acid to begin the polypeptide chain. The process continues until a stop codon with a particular sequence tells the ribosome to stop. The polypeptide is released and will be processed and folded into protein, the protein that was coded for in the gene on the DNA. The cell cycle is the name given to the process by which somatic cells replicate. The cell spends most of its time in interphase. It is during this time that the cell prepares for replication by creating the materials needed and replicating its DNA. The cell then enters the mitotic phase of division. In prophase, the chromosomes condense, spindle fibers form, and the nuclear envelope breaks down. In metaphase, the chromosomes line up at the metaphase plate. In anaphase, the sister chromatids are pulled toward opposite poles, and in telophase, the chromosomes decondense and nuclear envelopes form around each. During cytokinesis, the cytoplasm divides and the result is two cells that are nearly identical to the parent cell. Certain cells in the body are able to differentiate, that is, specialize into specific cell types. These cells are known as stem cells. All of the somatic cells in the body contain the same DNA, but differences in gene expression dictate what type of cell will result. Transcription factor proteins regulate this gene expression, leading to the development of specialized cell types. Finally, this slide shows two examples of differentiation pathways. On the left, we see that a hematopoietic stem cell differentiates into the different blood cells and immune cells. On the right, we see that a totipotent embryonic stem cell differentiates into cells that will form the endoderm, mesoderm, and ectoderm structures. These cells further differentiate into specialized cell types, as shown here at the bottom of the image. This concludes our review of Unit 1. Thank you.